Okay, hi guys. Today we are going to be looking at drug testing. And let's get the sillies out now. Okay, because we're talking obviously about legal drugs, medicines, and that kind of thing. So the first thing that we need to talk about actually is firstly, what is a drug? And that is just something that alters how your body works. I'm going to put cells, in fact, how your cells work. Okay, and that's a terrible S, but I apologise about that. So what makes a good drug? Okay, so what makes a good drug? Well, four things. Okay, let's draw a few pills there. Okay, so it's got to be effective. So if it's not effective, then obviously there's no point in using it. So if it doesn't do what you like, if you take a couple of paracetamol and the paracetamol don't do anything for your headache, then you may as well have not taken them. Plus also, I mean, drugs do have side effects, even paracetamol, okay? So you'd be doing something to your body which is just harming it rather than actually doing anything good. That brings me on to the second thing. They need to be safe. Okay, so obviously what we don't want is anyone to turn into a little skull and crossbones. Okay, so let's just draw them in there. Again, I mean, all drugs have got risks. Okay, any medication that you take will have an effect on your body, and sometimes that effect is not quite what you expect it to be or what you want it to be. So, for example, I've spoken about paracetamol. Your body can process paracetamol pretty well, but it can also harm your liver. So you can't take too many of them, which is why there is a dosage on the packet. However, obviously the positive side of that is that it does what you want it to. It gets rid of the pain in your body. Okay, so some drugs though, and particularly if we're looking at some of the more serious illnesses, if you've got, for example, cancer. Cancer is not really chemotherapy is not really in terms of drugs, but you're putting chemicals inside your body, and they are actually harming your body cells as well as the cancer cells. But the risk is deemed to be less bad than the actual disease itself. But that's for a different topic. We'll come to that later. Okay, they also need to be stable. Okay, so I'm going to draw a little. Can you guess what it is yet? Okay, I'm just going to draw a little stable. Put a horse inside it. That looks a bit more like a crocodile, but I'm not that bothered. It was a very quick drawing. Okay, there's a stable. So yeah, they've got to be. They mustn't break down. If you buy a packet of this new painkiller and then in a couple of days' time they've broken down into their component molecules, then there's no point having bought them because they've broken down. They also need to target and clear your body. Okay, so that means I'll draw a little target and clear. So that means your body's got to be able to process them and get rid of them quickly. They've got to target what you want it to do, the organ, the pain receptors, anything like that, and then you've got to be able to get rid of the resultant chemicals easily. So I've put a little poo there. Okay, so that's your four things, to make that makes a good medicine. Okay, they've got to be effective, safe, stable, and they should target and clear your body easily and quickly. Right. How long does it take? How long does it take to get a new drug from the first part of testing from choosing what you're going to do to it being in the doctors or in the pharmacists. Well, actually, that's quite a long time, guys. I don't know if you've got any ideas about this already, but it is 12 years. Okay, 12 years is how long it could take. Now, that's different from vaccines as well, because obviously every year we get a new flu vaccine to treat the new flu, and that will take less time than 12 years to produce. But that's like the longest amount of time really is 12 years, which is down. It used to be something like 25 years, I seem to remember. The other thing is, how much is it going to cost to produce our new drug? Well, that is a really good question. I wonder how much you guys actually think it would be. It can actually be up to 17 billion pounds. Okay, which I think is quite a shockingly large amount of money. Now, this also might give you the idea of why drug companies cost so much. Market their drugs and how much they cost. Okay, the price is quite high. I realise I'm rambling there a little bit, but the price is really high there, and that's because it actually costs them an awful lot of money to develop these drugs. There's an awful lot also of negativity about drug companies, and sometimes rightly so. But I think you've got to also remember that drug companies a lot of the time are making drugs to benefit everybody else. And yes, there is an aspect of profit in there, but that's actually kind of the society we live in. Now, I realise we're kind of getting into a little kind of political or philosophical context there, so I'm going to knock that on the head, and we're going to start looking at how to test for drugs. Well, you have to go through several stages. It's not just a dead straightforward process. So we'll start obviously with step 1. And you might be able to work out that in step 1, what we have to do is decide what we're actually going to manufacture. What's our condition? Okay, so we're going to identify our condition. Okay, that we're going to fight against. We're going to manufacture our drugs against. So we're going to, I'll draw some little bacteria in here. Okay, so we identify that there's this horrible bacteria, and we need to make something that will get rid of it. Okay, so there they are. Obviously, they're going to be coloured green because that seems to be the colour to colour bacteria in. So we've identified these guys, and what we also then need to do then is we need to use lots and lots of different medicines, different drugs, some that we've already got on the shelves maybe. And again, if you look at the coronavirus outbreak, that's actually what they're doing right now, is looking at potentially repurposing current chemicals, okay, that may have an effect on the coronavirus. Okay, just give them a little bit of colour. And we will use these to attack our pathogen. Right, so there we go. Okay, there we go. So we've got a load of different drugs there. We're going to test them on the pathogen. Is that enough? That will certainly start to work out whether what we're doing is effective. Okay, but the other part of it is, the other main part of it really is it safe? So we're also, not only are we going to chuck that at the bacteria, but we'll also chuck that at some human cells. Okay, let's draw some human cells in there. Those are obviously the good guys. We'll colour them in pink. There we go. And then that will identify whether what we're doing is safe. Okay, so I've drawn 5 test tubes there, but actually in reality we're going to be testing 5 to 10,000 different drugs. Okay, that is a huge number. Okay, so let's test different drugs. Test different drugs. Okay, so step 1, we're testing efficacy. Okay, that is, are they effective? Do they work? And toxicity, are they safe? Okay, so once we've tested all of our 5 to 10,000 drugs there, what we're going to then do is move on to step 2. Okay, which comes straight after step 1, as you probably know. Step 2. And step 2, okay, is, some of you may think of as controversial, but it is animal tests. Okay, so we've tested on human cells and we've tested on the bacteria to check whether it works. And now we're going to test it on a whole animal. Okay, and again, our little test subject is going to be a little mouse. Okay. Let's give him a cute name like test subject A164. And he's going to be ill. So he's got the disease. Okay. And he's not feeling very well. That's a little tail, pink nose, pink ears. Right. So we're going to inject him with some of our drug. Okay. And we're going to find out whether he gets better, which we hope he does, obviously. Or there we go. Or alternatively, obviously the one we don't want to think about is he going to die? Right, so this will give us a lot of information. Now, bear in mind that before we've done this, okay, we have tested on human cells. Like mammal cells are relative. I'm going to probably get a load of trouble if a load of people from zoologists or whatever get hold of this, but mammals, their cells tend to be fairly similar. Okay, so there's not a huge difference between them. And so if we've tested them on these human cells and the human cells have been okay, then chances are we're going to get more of this than this. Okay, at least that's the hope. So again, this is working for looking at efficacy and toxicity. Okay, toxicity. So we're checking, is it effective? Is it safe? This is the whole purpose of all of these trials. Okay, and then we're going to get, it's probably going to be more a choice of which of these drugs is most effective rather than, well, which ones have killed fewest creatures. And then we're going to get on to step 3, okay, which is a bit more in depth. And we're going to break that down a little bit more. Okay, so step 3. Step 3 is human trials or clinical trials. Clinical trials. Right, I should also add, because I forgot to say this just a second ago, these are also called pre-clinical trials. Okay, pre-clinical trials. So in here, obviously what we're going to do is we're going to give our patient the drug. He's feeling poorly. Obviously I've done him in a bed because he's in hospital, but it's mainly so I don't have to bother drawing a body, if I'm honest. There we go. Okay, so there he is in the bed. One of those little funny blanket things on like that. And we'll give him the drug. Like that. Okay, and we're going to see, does he get better? Gosh, he's got a massive pair of ears there. I didn't quite mean to do that. Is he better? Like this guy, or is he not better? And is he still going to be poorly afterwards? He's got massive ears as well. So the guy gets better. There he is. Bright pink. And this guy's eyes don't look very good either. He actually looks like I've turned him into a zombie. Let's hope that none of our clinical trials turn anyone into a zombie. And again, we're checking for efficacy and toxicity. Toxicity. Okay, you can tell which words I struggle with. Right, so step 3, the clinical trials. Let's break that down into a bit more detail. Okay, because in step 3, we actually get 3 different steps. Okay, the first one, okay, is where we have small doses. Okay, I'll just draw a tiny little vial with a drug in it, and we are checking whether the drug is actually safe for humans. Okay, that's the first part. We should, after having been through human cells testing and testing on animals, by the time it gets here, we should be fairly clear that it's safe. But we do need to check that. There have been some clinical trials in the past where things have gone a bit wrong. So what we need to make sure is that, yes, it is definitely safe. So we test people with just a small amount of the drug first. Once it gets through that stage, we then test it on larger doses. Okay, but with a small cohort. Okay, that means a small number of people. Small cohort. I'll put in brackets number, not hashtag, number of people. Okay, so we can just maybe have a bit of a and a couple of stick men just to make it clear. Okay, so then once we've got through that part, we can go on to doing step 3, okay, which is larger doses again. And a large cohort. Okay, so we'll draw again. About this time, I'll do lots of people. I'll make that effectively look like a massive number of people just by drawing some circles. You could have. There we go. And again, we'll colour that in green. So here, as I say, we're checking that it's safe. Here, we are checking for its effectiveness or efficacy. Okay, is it effective? Okay, and here, actually what we're testing for here is to check the dosage. Okay, how much do we need to take? So, for example, paracetamol, again, I'm going to keep going back to it because it's a very simple one, we all know it. You calculate the dose in hospital, you'll notice if someone needs paracetamol, they're actually calculating the dose of paracetamol according to their mass. So, whereas obviously the stuff that we buy in the shop is already dosed. And if you're, a kid, you're assumed to be a lot smaller, so you get given 1 or half or, 5 mils of Calpol because it contains less of the drug. Because, as I've said before, that some of the side effects can be quite dangerous. And for adults, you'll maybe get 2 pills. But that, again, it depends on your mass. And that's what they do here in step 3 of these clinical trials. Okay, is that they check for how much is safe to give people and how much they should give people. The other thing that they do in order to work out is efficacy here, okay, I'm just going to draw a little loop around here, is they do something called a double blind trial. Now, there's a couple of things I need to first teach you. Number 1 is what a placebo is. Okay, a placebo is a dummy drug. So they'll give you a pill, it'll probably be full of sugar, okay, and that's a placebo. But the doctor, in a double-blind trial, the doctor and the patient, neither of them, they don't know if they're getting a placebo or the actual drug. Okay? They don't know what they're getting. Because they've found that actually, if the doctor knows, then they can kind of communicate that almost kind of subliminally to the patient. So the patient starts to feel better because the doctor knows that this is the drug that should make them feel better. Okay, and this is called the placebo effect. So, and also some people who take the placebo thinking that it is the drug start to feel better as well. But that's only a low percentage of people, and you can cancel that out of the study. And that's why they do the double-blind trial. Now, after all of that's done, okay, then and it's passed everything, it's been approved, then we can get our drug out on trial. I'm going to call it Brain-O-cetamol. Okay, we can get that out to the public, okay, and we can start helping people feel better. So that is a brief description, a bit of dual coding on drug trials. I hope you found it useful. I hope you've made the notes. I hope I've got everything right. If I've not, then let me know. And yeah, take care, guys. Wash your hands.